So I'm really, really excited to host this chat with one of my heroes. Not too many people have worked as successfully as Sue has across industry, academia, and nonprofit, and worked with and survived working for and with Art Levinson, Mark Zuckerberg, Bill Gates, Warren Buffett, and the list goes on and on. So Sue was born in Napa and then joined the faculty here at UC, and then you were loaned to Uganda?
Yeah, when I was on the faculty at UC, I had trained in internal medicine and cancer and did HIV like everyone in the early 80s at UCSF. And so when Rockefeller Foundation got worried about the notion of heterosexual transmission of HIV, which in those days was thought to be a myth, it was, remember the 3 H's? You had to be homosexual, hemophiliac, right? Or Haitian, which is bizarre when you think about it now. Those were the 3 H's, but when heterosexual transmission was first reported in sub-Saharan Africa, Westerners thought that was probably people who were embarrassed to confess that they were gay. And so Rockefeller talked to UCSF, and UCSF loaned me and my husband, an infectious disease doctor, to Makerere University, and we lived in Uganda for 2 years, and I ran the Uganda Cancer Institute.
Wow. And so tell me.
Why not?
Yeah, exactly. Tell me a bit about your experiences there and how you were able to, the work that you did there kind of positioned you to come back and have your career in oncology.
So there was a couple things that I think, and I do a lot of mentoring, so there were 2 things about that experience that were really profound to me that still inform how I think. One was that we had tiny resources given the challenges that we were taking on. And so one of the things I learned to do in Uganda was to do SAS programming, the underpinnings of how you do biostatistics. I didn't have a statistician. And I didn't have a TV, I didn't have movies, I didn't have newspapers, so I read the entire set of volumes of this statistical programming tool that people use for biostatistics. And I would have never done that in those 2 years as a faculty member had I not been in Uganda. And then later, when I went into pharma and biotech, I knew the underpinnings of the statistical programming that drives how we do clinical trials, because I needed to, because I needed that tool when I was there. So on the technical side,
you went to Uganda for your degree in computer
science. I got my coding skills in Uganda, unusually, but more profoundly, the sense of having privilege, that everything that I had experienced in my life up to then was so profoundly different than anyone I interacted with in Uganda, and the sense that if I didn't do something to give back or make a difference, I would be ashamed of myself. That was, for me, became how I made career choices. Every minute after that was this sense of how lucky I am and was, and that most people in the world, when they woke up, didn't know where the next meal would come from, didn't have access to clean water, electricity, things I took for granted before that. And that was after going to medical school, going to get a public health degree. All these experiences I had were from a rich world experience. So it was a complete shift in how I thought. Oh, and by the way, I ruined my academic career.
Yes.
I did. When I came back, it turns out that the chief of medicine had changed while I was gone, and I didn't have a job when I came back.
So now
which was kind of disappointing.
So now you've got a computer science degree from University of Uganda, and you come back, you're jobless.
And I was jobless, but I knew that I had a skill that was useful for actually paying the rent and having a job. So I went into practice and became a practicing oncologist.
Now, you were a practicing oncologist for a couple years before you then made another move into big pharma biotech. Tell me a little bit about what the impetus for that move was, and maybe you can talk a little bit about the companies you were at.
So the impetus was that I went to Bristol Myers Squibb. I was a trailing spouse. They hired my husband to work on the second antiretroviral ever approved for HIV, and he said he wouldn't come without his wife.
What a romantic.
He's a good guy. And this is where the programming story comes back. So there was a little tiny drug at Bristol Myers Squibb called Taxol, and it had been approved for ovarian cancer, and they were about to work on the approval for breast cancer. And so they were very, very, very busy and had to find a place to put me because I was the trailing spouse. And so I went to work on the Taxol team doing drug safety, which at the time was where people went who weren't fancy enough to work on the efficacy and the more sexy parts of product development. But I knew how to program and I knew statistics, and so it worked out really well. And after a year, they made me the project team leader for the Taxol team.
For people in the audience that don't know Taxol, maybe you can tell them how big a market it is, how big a drug it's become for cancer.
Well, what was great, and that was the first year I was in industry, the nice thing about that was that it became the #1 cancer drug in the world, and Bristol Myers Squibb became the #1 cancer, was the #1 cancer company in the world. And so we got Taxol approved for breast cancer in the US and in the EU, and it went really well. So much so that the company and our team, the Taxol team, became known, which led to me being recruited to Genentech.
So now you go back across the country,
Yeah.
come back here, and you just had started such a great franchise at BMS.
Yeah.
And you're now going to Genentech that's doing a total new modality using antibodies instead of small molecules for drugs. Tell me about that change and how you made that decision.
Well, the critical part of the decision, there were two critical parts of the decision. The most important one was that I met Art Levinson. And Art at the time wasn't the CEO of Genentech. He was the head of R&D. And Genentech had exactly 0 revenues in cancer at the time. They had growth hormone, the heart attack drug, and a tiny little drug for cystic fibrosis. But Art had this dream that they would be a cancer company because he was a cancer biologist and thought that the company had assets that we could use to become a cancer company. But I took on faith, having met Art, that the great science that Genentech was doing could translate into being a cancer company. But that was because he was so compelling and the science was so compelling, not because there was a lot of evidence of it.
Well, Genentech is the Google or the Facebook of biotech and pharma. They continue to recruit the best and brightest after so many years. Tell us a little bit about the culture at Genentech as you saw it evolve, and under Art's leadership as well.
Well, when I went to Genentech, there were 2,000 people, and which for a biotech company doing everything. So it was a fully integrated company doing sales, manufacturing, product development, which was really relatively small. And the same year that I went to Genentech, Art became the CEO, and he had some of the best characteristics of any leader I've ever worked with. One of the most profound ways that he led was that he had a vision, for example, that we would be a cancer company using this entirely new way to treat cancer, and that we would do that by using great science and not have what in biotech at the time was often magical thinking, that we wanted to be a cancer company or wanted to do something special, but that we would go deep. We told investors in 1995 and 96 that we would spend nearly half our revenues on R&D. And some investors were unhappy and left the stock.
Just again to baseline, what would most of the competitors spend in terms of revenue and R&D?
If they had double digits, that would be
Wow.
Extraordinary. So that was profoundly unusual. So all of the people who went to Genentech knew that Art was that committed and was willing to tell investors, don't invest in us if you are not capable of having us put that kind of resources against R&D in quest of turning this great science into a cancer company.
How did Art and yourself, as you moved along the ranks at Genentech, motivate the team and keep people motivated to work and stay at Genentech as opposed to leaving and going to some of the smaller competitors?
I guess the most important thing was the belief and the reality matched. So there were a lot of people who liked to talk about following the science, and the fact that was real, and that if you thought that we were doing something in the company that wasn't consistent with what the science showed you, you could argue. You could argue with me, you could argue with Art, you could argue till you're red in the face and pound your fist on the table, and it wasn't just acceptable, it was desirable that you had a voice and you used that voice. And the other one that I think was even more important for me building a team, and we had more than 50 cancer doctors by the time I left, was that we wanted to do the right thing for patients. And that if there was something that could lead to making more money, but that it wasn't the right thing for patients, we wouldn't do that. So a good example in the early days was on Herceptin, that the big market thing would have been to develop Herceptin for everybody, but it would be so much better for patients if we could narrow the market and make the drug better, which now sounds obvious,
Right.
and then was unusual.
That's sacrificing lots of revenue to make sure the patient experience is better.
Absolutely, and so the best employees, the best talent in the world wanted to work that way, but were deeply suspicious that it would be real. And so I think that was the powerful thing that Art enabled, was that it wasn't just a slogan, it was real, that you followed the science and you did the right thing for patients.
Now, Genentech had many of the names that we all know of now, Rituxan, Avastin, Herceptin, all were under your watch. How did you guys decide which drugs to push through and which ones not to? And maybe give us a couple anecdotes of some of those stories of where you were right or wrong and the like.
I'm still licking my wounds over the wrong ones. Well, hopefully for the entrepreneurs and the companies that are here, there's some things to learn about how you manage a portfolio. And it ended up being, in the 14 years I was at Genentech, the last 5 years I was the president of product development. And the most interesting part of it was managing the portfolio. So and I often hear Silicon Valley founders and entrepreneurs talk about optionality. It's kind of a jargony word, but I wouldn't have used it then, but I think I became the master of optionality. And the reason I use the word optionality is we would have a certain amount of money, and the decision-making tried to look at when we had to make a decision to spend more money. And so the maximum number of bets that we could afford before we had to say yes or no was the best way to manage the portfolio. And so if I could bet on Avastin, and I could make a small bet for as long as possible until I knew I had more Rituxan and Herceptin revenues to pay for the very, very expensive trial on Avastin, then I would take those bets. And we did 2 big bets on Avastin, one that was much more high risk, faster, and was negative. Avastin didn't work in late-stage breast cancer, but we had a bigger, more certain bet in colon cancer behind it. And so we maximized the optionality of a fast, quick win, which didn't work out, but we had already planned for the longer, better science, more likely to succeed win.
So when Avastin didn't pass for late-stage breast cancer, how did you manage the morale and the enthusiasm of the team? It must have been disappointed.
It was disappointing. Is kind of an understatement. We had there were, at the time, very few good options for women with HER2-negative breast cancer, and that was our target. And so first and foremost, we managed it by making sure that the most important disappointment was the most important disappointment, which is that we didn't have something new for patients. So we worked really closely with patient groups. We anticipated and always talked about it as a high-risk trial so that we were trying to manage expectations, so we didn't overpromise and underdeliver. With Wall Street, I think investors were disappointed, and again, we had backup strategies and a way to win even when that trial lost. And with employees, I always rewarded employees for a clear answer. And I think in product development, that's an under-recognized attribute. Our job, especially in clinical science and in medicine, truth exists, right? Our job is to ask and answer questions, and I always managed people and always rewarded people by getting a crisp answer, even if the answer is no. Because our job in product development, especially if you take appropriate risks, isn't always to get a yes.
Right.
It's to get an answer efficiently and cost effectively.
And empower folks to disagree with you.
And to have people say, well, next time, would you make that same bet? So we had a famous failure that, that's the one I'm still licking my wounds over, where I think we made a big mistake. We had a new drug, a potential new drug, nerve growth factor, for diabetic neuropathy, for the numbness and tingling that patients with diabetes get. And we so wanted to make something new for patients with diabetes, but we didn't have an effective placebo in the phase 2. We didn't have an effective blinding. So the patients who got the nerve growth factor knew they got the nerve growth factor because it caused a little bit of pain when you got the injection. And that unblinding misled us into thinking we had a new drug. And then the phase 3 trial was absolutely negative. We efficiently knew it wasn't going to help patients, so that was the right thing to do. But we went back and did a lessons learned and learned a lot about how you can be misled, especially with something like pain, when people know what they're getting.
Well, the Genentech culture, you're on the board of Facebook, and Art was on the board of Google and Apple, so it really is pervaded through the valley. So you moved from Genentech then, after Roche did the acquisition.
After Roche bought us.
Tell us about the next phase of your career.
So Roche bought the rest of Genentech in 2009, a squeeze out. I learned that new finance term, which I was...
About optionality.
Yeah, I'm learning all these things. They weren't in the medical school textbooks. But I can tell you, squeeze out is not what you want to be on the other side of. We tried to resist, but did the right thing for our shareholders. And so when Roche bought the company, UCSF recruited me back as chancellor, resuscitating my academic career. So I moved back to UCSF in 2009 and oversaw the life sciences. So it's just a graduate education, didn't have undergrads and only life sciences, and ran a big academic medical center at UCSF.
And there you worked on President Obama's task force to...
Yeah, I got a chance to do something really special. As chancellor at UCSF, given the background I had at Genentech, I was able to co-chair a National Academy of Sciences report on precision medicine, which was terrific and outlined a vision for how the kind of thing we did with Herceptin and what at the outset was called personalized medicine could be done at scale for a number of diseases, and led to President Obama's precision medicine initiative.
So I got to ask, have we seen any results from precision medicine, or is this going to be nanotechnology all over again?
Yeah, you should be skeptical. The dream of precision medicine, I strongly believe in. But I think there's two giant watchouts. One giant watchout is what you just asked, which is I hope healthy skepticism that only by bringing big data or sequencing to the table will solve all diseases. The deep biologic understanding and the challenge of making it real for patients and making it real to solve more diseases is a massive amount of work. So I think it's a great dream. I believe in the dream, but skepticism is warranted. The second problem with precision medicine that I've been working on now, and is even more exciting to me than what we set out to do in 2010 with the Precision Medicine Report, is the biggest threat of precision medicine, and that is that it further increases inequities in medicine. So precision medicine easily is narrow, orphan-like diseases at massive cost. Mm-hmm. So you have fewer and fewer patients treated with bespoke remedies. And that means that the rich get richer, people who have means get better and better medicine, and the poor get poorer.
The concern being that the diseases of the poor will be avoided and neglected by the large pharma biotech companies.
Right. And further broaden the, whether you're in a rich country or a poor country, further increase inequities in access to great health interventions. So I've been working really with great vigor for the last year on something that I'm calling precision public health. Mm-hmm. And that is trying to bring this world of big data, consumer monitoring, low-cost sequencing, and all of the promise of precision medicine to a world that thinks about interventions that a frontline healthcare worker can use or a public health department can use that are scalable, and think about prevention and much more increasing equity by using precision as an efficiency tool.
There's a big setback in the field with the negative press around Theranos. Do you have any opinion on that? I have to ask.
You have to ask. So I always start, I think there's two big lessons that I would take away from that experience. There's a lot more than two, but the two that I would put forward. Lesson number one is, and I loved that Chris in his interview talked about Facebook. When I first started talking to Mark Zuckerberg about going on his board, I pointed to the sign in his office that said, "Go fast and break things." And I said, here's the thing about my job. Whether I was at Genentech or UCSF, I love going fast. But it turns out when patients' lives are at stake, you don't get to break things. Yeah. Not okay. I mean, the consequences of being wrong when it comes to people's health and diagnostics are profound. So you can try and go fast, but you don't get to break things. That's lesson one. Really, really, really important. The other thing is, and I think this is under-recognized in our environment, therapeutics are hard. Therapeutics, I worked on therapeutics for 16 years in industry. Making new therapeutics is one of the hardest things I've ever done, but there's actually something harder than therapeutics, diagnostics. Diagnostics takes the therapeutic degree of difficulty and puts it on steroids. There's nothing more challenging than putting forward something and says, I can take a sample from you, right? I can take a sample of your blood, your sweat, your urine, whatever I'm sampling of you, and I can predict the future. That is wicked hard. Mm-hmm. And if you like statistics, and I love statistics, if you like statistics and you think about as hard as it was to make Herceptin, to increase someone's lifespan who's struggling with a really difficult cancer, that was hard. Right. Now I'm trying to predict the future with a diagnostic. The degree of why that's hard is so underestimated that every job I've had, whether it's at UCSF or now at the Gates Foundation, when I interact with my team on a therapeutic, we're like, okay, know what we need to do. Here's a target product profile. Here's what we have to prove. It's hard but straightforward. And I always say, let's get an efficient yes or no, right? Next milestone, let's get an efficient yes or no. With the diagnostic, I have longer conversations, harder conversations, because that ability to predict is essentially more profoundly difficult than anybody realizes.
You've been involved in the development of many of the cancer treatments today. We were talking in the hallway about where you thought early detection could play a role and where you thought, in your opinion, it wouldn't. Can you maybe expand on that a bit?
Yeah, I think the early detection question is part of my problem statement about diagnostics. So the only thing harder than predicting the future is the ability to predict the future when early detection means that if you have cancer and I find it early versus how I would find it if I didn't use my new test, what happens to you? And what's the consequence of being wrong? So early detection means that my intervention prevents something bad from happening to you. That means that my intervention is better than if I didn't find your cancer early and I found it by chance alone or self-detection or you feeling sick. We have found in breast cancer with mammograms and in prostate cancer with PSA, that is profoundly difficult to do. In colon cancer and in cervical cancer, we've increased survival with early detection. In all the other cancers, it's been a high bar to do that.
Does that mean that women shouldn't do mammograms, or?
It doesn't mean that women shouldn't do mammograms, but it means that a lot of what we believed was helping women with mammograms was detecting cancers that probably she would have done well with even if we hadn't detected them early.
Wow.
Yeah, it's really annoying because the dogma and the wish that we all have that would help women was so powerful and so strong, or that we would help men through PSA testing is so powerful and so strong because we want to help. And the reality is it's actually quite difficult to help by intervening early.
We're going to run out of time. This is great, but last phase of your career, or
Yeah.
maybe your next phase of your career.
Current.
Phase, right. So tell us about what made you go into the nonprofit world and what some of the things the Gates Foundation is doing and how maybe you can work with some of the companies here.
So 2 years ago, Bill and Melinda recruited me to go to the Gates Foundation, and so I left UCSF and traveled up to Seattle. And what I'd want people here to know about the Bill and Melinda Gates Foundation is it's an amazing, ambitious agenda. I'm sure you'll hear more about it from Bill tomorrow. And one of the tactics that we use that I'm extremely excited about is what we call program-related investments. So unlike a lot of not-for-profit work and a lot of classical global health or global development work, we believe that the private sector has tools, talent, ambitions, assets that we want to put to use in the not-for-profit sector. So we've come up with a set of tools, volume guarantees, tiered pricing, equity investments, that with a charitable intent, we can invest in companies. Some companies have allowed themselves to meet
you're now going to get hounded as soon as you walk
I'm
off
happy
the
to be
stage.
hounded. I'm open for business.
Okay.
But what we love is companies that have an aspiration to make money and that we can help them early on fund R&D, fund product development in things that work for the poor.
I'm going to try to help you out so you don't get mobbed.
Okay.
Everyone here, maybe some more specificity. What specific areas are you guys looking at?
So we care a lot about infectious diseases. So specifically infectious diseases that cause a big global burden of disease, HIV, TB, malaria, neglected tropical diseases, everything from schistosomiasis to onchocerciasis. We're very interested in new ways of thinking about pandemic preparedness. So two things that are part of our dream are diagnostic formats that can be turned around quickly, and vaccine or therapeutic formats that if we knew the antigen of interest, we could go fast. So those are things we're particularly interested in investing in. We're also investing in agriculture and financial services for the poor. So we're very interested in anything that we can do that goes to low resource areas, that gives smallholder farmers better tools to be more productive.
full circle from Uganda going back. We're out of time, but I got to ask the question because everyone's dying to know. Did you read Vinod's paper on 20% doctor needed?
I did, and I'm a little nervous as a physician. Here's the thing that I think is so I love the paper. I love the thinking.
Oh, now I'm gonna live with that.
Sorry. Now you have to live with that. Here's what I think is so important. A lot of what we need to solve to make the world a healthier, more equal place, which I think about every day now, isn't going to be solved by doctors. It's going to be solved with much more of a self-service model that we use in our life for a lot of other things. So I strongly believe in a more self-service model. I think some of the things that were talked about earlier on this stage give huge opportunities for more self-service. But also, I love the concept of every healthcare provider working at the top of their license. Mm-hmm. And in many of the places I work, the top of that license might come with a third grade education. So to have that healthcare provider or that community healthcare worker work to the top of his or her ability, and then that physician, that nurse, that medical officer work to the top of their ability is a much more cost-effective, more productive way for us to deliver health services.
That's great. Well, we're out of time. Can we please thank Dr. Hellmann for her speech?